Hello and welcome to my video today. My name is Corey Heisey, and today I'm going to go over a research paper that I found interesting out of Southeast University in China. It's about machine learning and neuroscience, and let's get into it. Look at this output from an AI. Prefrontal hyperconnectivity, limbic dysregulation, default mode network, overintegration, the network profile supports MDD diagnosis certainty. Hi, that's a language model rendering a brain scan and writing a clinical report in plain language, not just making a prediction, explaining it. And it's doing this better than any brain AI that came before it. Today we're breaking down exactly how that's done. Before we get into the method, we need to build two pieces of intuition. What idea actually looks like, and why existing AI struggles with it. When you go into an fMRI scanner, the machine measures blood oxygen levels in your brain across 90 different regions every second or so while you're lying still. Those measurements produce what's called a BLEG signal, a time series for each region. Here's the key insight. If two brain regions tend to activate at the same time, they're considered functionally connected. You compute a Pearson correlation between their series and their correlation between the edge weight in a graph. After thresholding to keep the strongest connections, you end up with a sparse brain network where nodes are brain regions and edges represent how strongly connected they are. This is the graph that gets fed into a machine learning model. Now, graph neural networks are really good at pre-processing this kind of structured data. They pass messages between connected nodes, aggregated neighborhood information, and eventually output a classification. Is this person healthy, or do they have an autism, depression, or ADHD? GNNs have been the go-to method, and they work, but they hit a wall. Section 2. The problem with GNNs can't solve. Problem 1. The graph is sparse. Most brain graphs only have a few hundred meaningful edges out of over 8,000 possible connections. The pre-processing pipeline throws most of the signal away. Problem 2. And this one's subtle. The model has no domain knowledge. When a GNN processes the graph, it sees region 12 connects to region 47 with strength 0.6. It does not know that region 12 is the amygdala. It does not know that the amygdala hyperconnectivity is a documented signature of major depressive disorder. It doesn't know any neuroscience at all. Think of it like handing a brilliant statistician a brain connectivity spreadsheet, but never giving them a neuroscience textbook. They can find correlations, but they can't leverage decades of published research about what those correlations mean. That domain knowledge is trapped inside the neuroscience literature, which conveniently is exactly what large language models have been trained on. So the question becomes, how do you get a large language model's understanding of neuroscience into a graph neural network? Section 3, the BLEG method. This is what BLEG solves, and the approach is clever. They don't try to replace the GNN with a language model. That would lose all the graph structure and fine-tuning. A full LLM on a few hundred brain scans would be catastrophically expensive. Instead, they use LLM enhancer. 3 stages. Stage 1. Use an LLM to generate neuroscience descriptions for every brain graph. Stage 2. Fine-tune a smaller language model on those descriptions and train that GNN alongside it for rough alignment. Stage 3. Fine-tune just a small adapter on top of the frozen GNN, using an alignment loss that pulls the GNN's prediction toward the language models. Let's go through each step in detail. Section 4. Stage 1. LLM text generation. The first challenge is, how do you describe a brain graph to an LLM? An fMRI graph isn't text, it's a matrix of numbers, so the authors convert it. They represent each edge as node I, strength, node J, so a strong connection between the superior frontal gyrus and the precuneus might look like node 3.8, node 28. They combine the structural representation with node features, the mean activation value for each region, and then write a prompt that tells the LLM. You're an experienced neuroscience researcher. You're looking at fMRI connectivity data from a patient in a BLEG dataset, where the task is to classify autism versus healthy control. The brain regions are named as follows. This is a graph. Please analyze the pattern, output a JSON with analysis, key features, and your conclusion. They're using DeepSeek Vision 3 as the LLM. For every one of the 47,060 brain graphs across their public dataset, they run this prompt and get back a structured JSON response. Here's an example from autism dataset. High connectivity in prefrontal regions, strong temporal lobe connections, subcortical involvement. The observed hyperconnectivity is prefrontal and temporal regions suggest atypical neural integration in ASD. That's not a hallucination. Those are real documented signatures of autism in the neuroscience literature. The LLM knows this because it trained on papers that describe exactly those patterns. They also run a quick QWQ 32 billion parameter judges each response. Low quality one gets refined. The results in 47,060 brain graphs, each paired with a high quality neuroscience description. Section 5. Stage 2 is where the knowledge transfer actually begins. The team takes a smaller language model, GPT 347 million parameters, and fine-tunes it all those graph text pairs from stage 1. Here's the problem they face. There's a fundamental gap between graph representations and text representations. They live in a completely different high-dimensional spaces. To bridge this, they take inspiration from Lava, the visual instruction tuning paper, and feed both the GNN's node embeddings and the text tokens into the language model together. The GNN encoder and the LM are both kept trainable during the stage. The LM learns to generate the expert neuroscience descriptions given both the graph and the question. The GNN learns to produce embeddings that roughly align with the text embedding space. This is called coarse alignment. The graph and text representation are pulled toward each other, but not precisely. After instruction tuning, two things exist that didn't before. First, a fine-tuned BioGPT that understands the connection between brain graph structure and clinical neuroscience language. Second, a GNN encoder whose representations have been shaped by the task of describing brain graphs in clinical terms. It's exposed to neuroscience knowledge through the back door. But the fine-tuned grained alignment, the precision that actually drives performance on downstream tasks, that comes from stage 3. Section 6. Stage 3 is where the real magic happens. Both the GNN encoder and the BioGPT are now frozen. Their weights don't change. A small trainable adapter, a 2-layer feed-forward network, is added after the GNN. This adapter is the only thing that learns in stage 3. Here's how it works. A brain graph passes through the frozen GNN and the adapter producing a classification logic vector, especially the GNN confidence distribution over possible diagnosis. That same brain graph also passes through the frozen LM, which produces its logic vector. The language model's confidence distribution, the alignment loss in the MSE between these two normalized vectors, in plain language, make the GNN's confidence profile match the language model's confidence profile. The total loss is L cross entropy loss plus alpha times alignment loss, where alpha controls how much the LM's opinion influence training. This is elegant because at inference time, when you actually diagnose someone, you only need the GNN. The LM never runs. All that domain knowledge has been distilled into the adapter through training. The ablation study confirms this in the crucial piece: remove the alignment loss and accuracy drops across every dataset. There's also a theoretical response in the paper. Using mutual information theory, they prove that a GNN trained with LM alignment captures strictly more information about the downstream labels than a GNN trained without it. Section 7. Results. Let's look at what this achieves. Across 4 public brain network datasets, autism, ADHD, gender, classification, depression, BLEG outperforms every single baseline, including specialized brain GNNs and prior LLM GNN hybrid methods. On the Human Connectome project dataset, That's the gender classification task. BLG hits 71.21% accuracy versus 64% for a vanilla GCN. That's a 7% point jump against the prior state-of-the-art brain GNN method. Contrast pool BLG improves by 3 percentage points. But the results I find most interesting is the few-shot experiment. They gradually shrink the training set from 70% of the data down to just 10%. BLEG consistently maintains its lead over both brain GNN and brain NPT at every training size. This is important. The LLM's domain knowledge acts as a form of regularization, even when the LLM's domain knowledge acts as a form of regularization. Even when labeled data is scarce, the model has internalized something about the brain connectivity patterns mean. The prior knowledge doesn't disappear when training labels are limited. And quantitatively, when they visualize which brain regions the model focuses on, for autism, it highlights the precuneus, amygdala, and superior frontal gyrus. These are consistent with what the neuroscience literature says are ASD biomarkers. The model isn't just getting the right answer, it's looking at in the right places. Section 8, limitations. Now let's be honest about what this paper doesn't solve. Accuracy numbers sit in the 65 to 75% range. For a clinical diagnostic tool, that's not there yet. The paper correctly positions this as a research contribution and assistance tool, not a replacement for psychiatry. But it's worth saying clearly, you would not deploy this to diagnose patients. There's also a sensitivity problem on the ADHD dataset. Sensitivity of 41% means the model misses roughly 60% of actual ADHD cases. That's a clinical concern that the paper's aggregate accuracy number obscures. The LLM text generation is the weakest point in the pipeline architecturally. If the LLM writes something factually wrong, and the LLMs do hallucinate, even in medicine, that bad information gets into the training data and propagate through all three stages. The refinement step helps, but it's not a solved problem. Finally, the method is tested only on fMRI data, which this generalizes to structural MRI, EEG, or multimodal data is unknown. Section 9. Implications and conclusion. So let's zoom out. BLEG is at its core a specific answer to a general question. How do you inject symbolic linguistic knowledge into a geometric structural model efficiently and cheaply? The answer here is use the LLM to label your unlabeled data with the domain knowledge. Train a cheap student model on those labels and then distill the student's knowledge into your target model through an alignment loss. This is a recipe that generalizes well beyond brain networks. The same approach could be applied to molecular graphs in drug discovery, to circuit graphs, chip design, to any domain where a GNN is the right model, but the features are sparse and a large pre-trained language model has relevant domain knowledge. For practitioners building a graph ML systems, this is worth stealing. For researchers in computational neuroscience, the most exciting part is the qualitative result. A fine-tuned 8 billion parameter model reading a brain connectivity matrix and producing coherent clinical language. The path towards interpretable AI-assisted neuroimaging is generally exciting. If you found this video useful, the paper and sources are in the description. Let me know if you have any other papers you want me to analyze. Thanks for watching. Until next time, bye for now.